The next protein assay that we're going to be talking about is popularly known as the Bradford Assay. It gets its name after the scientist who developed it, Marion Bradford. This assay is based on the binding affinity that Coomassie has for certain amino acids under acidic conditions. Once the dye binds to the amino acid, its anionic form gets stabilized, which leads to a shift in peak absorbance from 465 to 595 nanometers. For you to perform this assay, you have to mix the stock solution of Coomassie to your lysate in a ratio of approximately 1,000 parts of dye to 25 parts of lysate. Once you've mixed the dye with the lysate, you let it incubate at room temperature for a short period of time, place it on a plastic cuvette, and then measure the absorbance at 450 and 595 nanometers. That's going to allow you to calculate the ratio between the two absorbances. So you just divide 595 absorbance for the 450 absorbance, and then you can interpolate your data based on the standard curve. To perform your Bradford assay, in addition of having your test samples, your standards, and your blanks, you're also going to need to have a spectrophotometer or a plate reader, depending on if you want to run your assay on plastic cuvettes or on a microplate. You're also going to need to either prepare or purchase the stock solution of Coomassie blue dye. The sample volume that you're going to be using is going to depend widely on two factors. Are you running your assay on cuvettes or on plates? And how sensitive do you need that assay to be? The higher the sample volume that you use, the more sensitive the assay is going to be. So if you're going to run this assay on cuvettes, you're going to need something between 30 and 1,000 microliters of sample. And if you're going to run it in a plate, you're going to need something between 5 and 150 microliters of sample per point. Now keep in mind that is not including those 3 technical replicates that we always need to prepare. Now, while Coomassie binds to several different amino acids, it does have a preference for arginine. So this assay is not recommended to be used in samples that are known to have low levels of arginine or that have proteins that have a small molecular weight. In addition to that, Coomassie also has a tendency to precipitate. So this assay is also not recommended to be used in proteins that are glycosylated or that are transmembrane proteins. This assay also doesn't perform very well in the presence of detergents or salts. For a complete list of reagent compatibility, please refer to the datasheet of your kit or to the references in the summary of this course. The Bradford assay is neither accurate or sensitive unless you use a very high volume of sample on each point. Its dynamic range is going to vary between 100 to 1,000 micrograms per milliliter to 15 to 100 micrograms per milliliter, depending on the ratio of Coomassie and sample. However, that assay is pretty quick, it's high throughput, and it is cheap. When you're running your Bradford assay, don't forget that the Coomassie blue dye tends to bind to a lot of different surfaces. So it's probably a good idea to use a plastic cuvette or a plastic plate that you can easily dispose of. Because Coomassie tends to precipitate, it's probably a good idea to also use a solution that is commercially available to reduce your chances of running into precipitation. If you're performing your assay on plates and your plate reader doesn't have those specific wavelengths that we mentioned before, you can use the range of 570 to 610 nanometers to read your plate without compromising sensitivity. Talking about sensitivity, if you need to increase the sensitivity of your assay, remember you can always decrease the ratio of Coomassie dye per sample volume to increase your assay sensitivity. Please don't forget to watch our best practices lesson from this course, since it covers a lot of important information on how to prepare the standard curve and how to analyze your data.